I want to welcome you to the showcase session. This is designed to give an opportunity and interact with experts in precision genomics. My name is Kathleen Barnes. I'm SVP of Population Health and Precision Medicine at Oxford Nanopore. Now, each of our speakers are going to present a brief overview of their work and their interests. Then they'll join me for a panel discussion where we'll be inviting you to ask questions and get your opinions. Because this is an on-site session, we'll be taking questions from the floor, but we won't be taking questions online. So at this point, I'm going to ask my speakers to come to my first speaker, Prasad Sarashetti. Prasad is an experienced bioinformatician. He currently serves as Senior Research Officer at the Genome Institute in Singapore. Here he spearheads efforts in telomere-to-telomere genome assembly and pangenome construction for the Asian Reference Genome Project. So his talk is entitled High-Quality T2 Assemblies with Oxford Nanopore-Only Data, a Singaporean Case Study. So without further ado, Prasad, take the stage.
Yeah. Thank you for the introduction and giving me this opportunity to share our results. Yeah. So here I'll be presenting how we have generated telomere-to-telomere assemblies only using the Oxford Nanopore-only data. So just to give a very brief introduction, on what a de novo assembly is, it's a computational process of generating a consensus using a read data set. So taking a diploid as an example, so we have two different methods. One is a haploid version of the genome where we merge both the haplotypes and have a single genome. But with the technological advancement, now we are able to distinguish those two haplotypes and we can construct truly a diploid genome where we can have a maternal and a paternal haplotypes separated. So needless to say, there are many applications for the de novo assembly process. For example, there is an organism which we don't have any references. De novo assembly becomes the first step to understand what this genome is really made up of. So now coming back to the human genomics and the references. So nearly after the first genome, it took nearly 2 decades to complete the missing 8% of the genome. That could only be possible when we have the advancement in the long-read technologies. With the advancement of these long-read technologies, we could complete that genome from telomere to telomere. So I'm also happy to announce that we have recently completed our first Indian genome. Till date, that the draft will be available in next few weeks, and that genome is one of the highest deployed genome available till date. So we have achieved the QV around 82. So that's 1 error in 100 million bases. So the draft should be available in next few weeks. So now the question is, can we achieve such high quality reference genomes only using nanopore data? So the current trend or the most of the references that have been already published are generated using HiFi dataset in conjugation with ultra-long data. So, but we can do the same thing only using the ONT data. So as you can see here, we are comparing the traditional method where HiFi plus ultra-long data is used, plus only the HiFi data, ONT data. So for phasing, we are in the hybrid approach, we are using a parental data sets, but for the ONT only assemblies, we are using PORC data. So as you can clearly see here, we have used 3 different data sets here. As you can see from hybrid versus ONT, NG50 value clearly indicates there is an improvement in the contiguity of the genome. But though your NG50 values are not clearly significant, but when we compare the telomere-to-telomere contigs, you can clearly see ONT-only approaches produce a lot more telomere-to-telomere contigs than the hybrid approach itself. So the one which are mainly missing from the ONT-only approaches are acrocentric chromosomes, which are really difficult to assemble because of our rDNA clusters. So coming to the phasing efficiency of both the methods, as you can see, the phasing efficiency are really comparable. And same thing goes with the QE value. So the QE is one of the method of measuring the quality of the assembly. Since de novo assembly is a process of generating a consensus from the reads. So what we check here is how many k-mers from the reads you see in the assembly. So any k-mers that are present only in the assembly are due to your errors, consensus errors. So coming back to the next evaluation, we checked the k-mer completeness. So here what we check is how many of your k-mers from the read data sets are also seen in the assembly. So majority of the k-mers what you see in the reads should be also found in the assembly, indicating that you have constructed the complete genome. And the second one is, so we also check the structural variations. Since our assemblies are from the reads, we shouldn't be finding more of those structural variations. If we are finding more of the structural variations, then it means there is an assembly error. So as you can see from the hybrid versus ONT only, so you have a very less assembly errors that are made using ONT only compared to the hybrid approaches. Yes. Coming back to the question, which is very important for large-scale studies, where how much does the time and it translates into the cost of computation. So as you can see, the current approaches of ONT-only method utilizes one of the error correction step that is GPU dependent. So if we have to translate that into a cost, taking by an example of AWS buckets, so the GPU buckets are much, much costlier than the CPU-only approaches. So that would translate into higher computational costs of ONT-only approaches compared to the hybrid one. So as an alternate to this GPU-based correction, so recently there is a new method which was introduced in Hyphaesium. So this is a CPU-based only correction method. So as you can see, the CPU-based correction method from the Hyphaesium is still better than the hybrid approaches, that is only using Hi-Fi plus ultra-long. But it is as good as your Hero, which is a GPU-based one. But with the current version, it's still in an early phase. The Hyphaesium correction method is still being developed. So the major disadvantages what we see is it still makes a lot of misassemblies. And when we check, those misassemblies are mainly in the centromeric regions, which are very difficult to assemble and even correct. Yeah. So on an average, yes, we can generate reference quality assemblies only using ONT platforms. And with the current algorithm limitations, it can be slightly costly in terms of computational, but given the improvement is what we have seen, it is worth to explore this path for a large-scale projects. With that, I would like to thank my whole team. Yeah, thank you.
Prasad, thank you. Fantastic. We're going to move on now. I'm going to welcome my next guest, Francisco de la Vega, also my colleague. Francisco is Chief Technology Officer and Senior Vice President of Platform and R&D at Galatea Bio. He's a distinguished human geneticist and computational biologist. He's recognized for his expertise in clinical and population genomics and bioinformatics. He's also an experienced technical executive with senior roles at Applied Biosystems, Tempus AI, and Fabric Genomics, where he successfully brought genetic analysis products and genome sequencing into the clinic. Francisco's talk title is Uncovering Genomic Diversity in the African Diaspora via Long-Read Sequencing. Thank you, Francisco.
Thank you, Kathleen. I'm glad to be presenting here a little bit what we're doing at Galatea Bio. Well, first of all, in 2011, Carlos Bustamante, Esteban Gonzalez-Burchard, and myself warned that most of the genomic efforts in the world were very biased to European populations. And clearly, that bias can translate in gaps in translation into the healthcare system, but also in lacking information about what's going on in the rest of the world. Since then, when we reported this, 4% of the individuals in GWAS studies were non-European, and unfortunately, up to last year, that has not changed a lot. So 2 decades later, we're still having this problem of representation. And there's many impacts of this lack of representation. From the biopharma discovery perspective, that's where we are interested in Galatea, databases that are rich across populations in the world will likely to get us into novel targets. It could also help in biomarker validation and support the tolerability of new drugs. And we have now multiple examples of drugs, starting with PSK9, where alleles present in other populations have the discovery of a potentially new drug target. So clearly, doing diversity is good business for biopharma. So in Galatea, we wanted to enhance the catalog of genetic variation around the world and develop cohorts that can be used for biomedical research. So in collaboration with Kathleen and Oxford Nanopore, we started a project to sequence about 500 individuals from the Consortium of Asthma among African ancestry populations in the America, which is an NHBLI project for the last 11 years. Our goal is to expand the catalog of structural variations, in particular in population of African descent, potentially finding some structural variations that have impact in disease genes or pharmacogenes, and also better understand how local ancestry inference, which is important to better understand how these admixed populations come about and the impact of that in health, how long read and back read phasing can help in these efforts. This is just a PCA plot that tries to depict the kappa populations on the background on the well-known populations from the 1000 genome, and we can see with different colors the continental level ancestries. The black squares are our populations. You can see that these populations actually complement and expand the genetic diversity of what is known in the 1000 genomes and other collections. So we think they will add significantly to these catalogs. We are able to discover about 31,000 structural variations per genome. This is almost double what you can get with short read. So there's an obvious advantage of using long read technology in these endeavors. Also, when we compare our data to the recently published 1,000 genome project with Oxford Nanopore, we see 60% novel SBs. So these populations also add to those other efforts undergoing around the world. Currently, we found about 200 of these SBs impacting OMIN disease genes, so it will have definitely some relevance. And I'm just going to show you one example. This is the filaggrin gene. This gene is involved in the barrier of the skin, on the structural parts of that, and has been previously implicated in immune-related diseases such as atopic dermatitis, asthma, and other diseases where the barrier, obviously, plays a very important role. So the exon 3, which is the main portion of the gene, contains multiple repeats of the filaggrin structural protein, and it's very difficult to sequence with short-read technologies. So what we can see here is we identify structural variation, which seems to be relatively common, but it has been, up to this day, not really cataloged. We also can see that there are problems aligning this, even the non-delete haplotype to the reference genome. So obviously, we think T2T assemblies of these individuals will allow us to better understand the structure of this gene. And we are finding more interesting genes that I hope that in the future we're going to be able to report, but gives you a glimpse of where we can discover when we start studying diverse populations. Thank you.
Thank you, Francisco. Fantastic. Okay, up next, I'm delighted to welcome Yu Kang. Yu is a professor at the Beijing Institute of Genomics, Chinese Academy of Sciences. Her research centers on sequencing technologies, genome assembly, and genomic analysis. She led the successful assembly of the high-quality diploid genome of a Han Chinese individual, T2T YAO, achieving a quality that's on par with the T2T CHM13 reference. Yu's talk is entitled Approaching an Error-Free Diploid Human Genome. Thank you, Yu.
Okay. Thank you, Kathleen, for the introduction, and it's my pleasure to be here to share my recent work on how to polish deploy the human genome towards an error-free status. To achieve this goal, an accurate genome quality assessment is essential. In 2020, the NIH group has introduced Mercury, that is a chimer-based approach that check if all the assembly chimers are supported by sequencing rates. And this method has been used to assess the T2T CHM13s and the T2T YAO from our group and various versions of AG02. However, we noticed that this chimer-based approach became less reliable when the genome quality improves, particularly beyond Q70. And the false positives can often occur in the regions of low or zero depths of the element or rival race, such as in the degenerative repeats of A, GGG, or T CCCs, where the sequencing bias may cause the underrepresentation of these genuine sequences. More critically, some real errors can be missed, particularly around the homopolymers, if they do not introduce a new chimer or masked by the sequencing noise. This problem cannot be fully resolved even using a larger 31-mer. To address this, we developed an alignment-based evaluation where we simply define the assembly error as the inconsistency between the assembly and the reads aligned to it. Using data from multiple platforms, we can identify both structural errors and base-level small errors. And the key indicators include large indels, clipped alignments, and abnormal read depths. And the later one, the small errors can be further classified into homopolymer-related and non-homopolymer-related small errors. In our initial version of T2T YAO, this method have identified hundreds of times more assembly errors than the 21-mer-based estimation. With over 70% related to homopolymers. As for the structural errors, this method seemed to be more sensitive than the SNIFOS2, as these widely used SV detectors may require the annotation of tandem repeats for the precise SV coding in this specific region. However, these annotations are often absent for the new assemblies. After several rounds of polishing, we have successfully eliminated all the detectable structural errors, excluding those in the rDNA clusters. And this further improves the alignment of element and rival rays. This enabled us to correct more than 90% of the small errors. And now the QV of 21-mer have reached 85.28, approaching an error-free status. So here comes the summaries. The first, chimer-based evaluation alone isn't sufficient, especially when the Q21-mer exceeding 70s, and alignment-based evaluation are more accurate in such cases. And the second, ultra-long ONT rates are critical for fixing structural errors and further improve the short-read alignment and the correction of small errors. Finally, I would like to thank Professor Gao and the Linfen Central Hospital for providing the precious samples that tied to the great immigration in Ming Dynasty and the ruined capital of the legend Emperor Yao, which is a cultural inspiration behind the name T2T YAO. I would also like to thank to all my colleagues for their vital contribution. Thanks for your listening.
Thank you, Yu. Thank you. Okay, our final panel member is Ya Bediako. Ya is a Ghanaian immunologist and entrepreneur. In 2020, he founded Yamachi Biotech to increase the representation of African data in oncology drug discovery. He's a 2021 Callistus Juma Science Leadership Fellow, supported by the Bill and Melinda Gates Foundation. He's a fellow of the Ghana Young Academy and an affiliate member of the African Academy of Sciences. And in talking with Ya, the list is actually much more extensive than that. His talk title is The African Cancer Atlas, Leveraging African Data to Revolutionize Precision Oncology.
Thank you very much, Kathleen. It's great to be here. And Francisco has actually done most of my intro for me, so I can hopefully get through this quite fast. So really happy to be here to tell you about the work we're doing at Yamachi, trying to diversify precision medicine and ensure that, Oxford Nanopore says what you miss matters. I would say who you miss matters as well. I have no conflicts of interest. So 1 in 4 people at some point in their lifetime will develop cancer. Now, if you look at cancer mortality from a sort of an ancestry lens, you find that black patients, and this is in the US, are 40% more likely to die of breast cancer, twice as likely to die of prostate and myeloma, and pretty much any cancer you can think of. The mortality to incidence ratio among individuals of African ancestry is among the worst. Now, Francisco already showed this. Genomic data is incredibly skewed towards individuals of European ancestry. Much of the African data we have is actually from diaspora populations who are often admixed populations. So you think of people from the continent of Africa who've actually been sequenced, it's actually less than 1%. Africa happens to also be the most genetically diverse continent on the planet. So the idea at Yamachi is if you have a diversity problem in precision medicine and in biopharma, the solution actually resides on the African continent. And I often joke that the moment we invented sequencing machines, we should have moved them to Africa and started sequencing across the continent. We didn't do that. We have a bias, but we can now do something about it, and that's what we're hoping to do at Yamachi. What we've done at Yamachi over the last 4.5 years is build a comprehensive network of partners across Africa. Currently, we have a partnership presence in 9 countries in every region on the continent, hoping to expand that to 14 by the end of this year, and then we have a few ideas of where to go next. This gives us a clear access to cancer patients across Africa. So when you think of Africa, you think of infectious disease, typically. But Africa has one of the fastest growing rates of cancer anywhere in the world, and the mortality of cancer in Africa is almost worse than anywhere else. So there are lots of people with cancer in Africa, and Africa is very diverse, so by effect, you have lots of diverse people with cancer, which is what pharma typically needs. So the goal in Yamachi is how can we generate this data locally and then make it available to pharma to globalize cancer research. So what we did at the beginning of this year is we launched TACA, which is the African Cancer Atlas. Those of you who are aware of TCGA will be familiar with this concept to build clinical genomic databases. TCGA is a fantastic database, but it has the common Achilles heel in that it's 80% European or Caucasian, has about 9% African ancestry patients, but if you spread them across 33 different types of cancer, for certain types of cancer, you literally have a handful of individuals of African descent. So what we're doing with TACA is building a database of 15,000 genomes, paired somatic and germline genomes from people across the African continent, so from West, East, North, and Southern Africa. And the goal here is to build up a database that can completely, hopefully revolutionize the way we think about cancer treatments. Francisco gave a great job of pointing out why that is important in terms of which markers you identify, both at a diagnostic and a therapeutic perspective. Now, when you think about databases, they're nice, they're great as scientists for us to build, but it doesn't really help anyone if it doesn't translate to the clinic. Ultimately, what each of us hopes to do is improve the lives of people who have, improve the lives of people, in our case, people with cancer. So we're very interested in how we can take data we generate in a knowledge base like TACA and implement it in the clinic. And so this is where our partnership with Oxford Nanopore has come in. Progress is a pediatric project, a pediatric cancer project that we launched a year ago with support from LIFARC in partnership with ONT. And the goal is to sequence children with cancer in Ghana. We've partnered with the 3 facilities that treat children with cancer. So pretty much every child in Ghana who gets a diagnosis of cancer has an opportunity to be enrolled in Progress. And we've begun doing long-read sequencing of these samples. Now, 25% of the children we've recruited out of about 350 we have so far, have leukemia. Leukemia is a highly treatable cancer. In this part of the world, in Europe and in North America, almost 90% of children with leukemia are cured. In our part of the world, it's less than 30%, largely because they present late and they don't always have access to the right care. So what we're trying to do now is, can we leverage long-read sequencing on nanopore to improve outcomes in children with cancer? It starts off by understanding the disease. Hardly any children with leukemia in Africa have ever been sequenced. So we've begun doing long-read whole genome sequencing. This is just one example of a child sequenced at, these are 2 children. We're collecting samples at diagnosis and then also longitudinal samples after induction, so after certain rounds of chemo, and following them up over time. And as you can see, we're getting close to 30x coverage on a Promethean running on a P2 solo, and we're getting N50s of over 30, in case on average 34,000. So in fact, I know ONT is quite jealous of the data that we're generating in Accra. So all of this is happening in Accra, in Ghana, locally, and I think that is the idea about localizing it. This is just one example of a structural variant that we found, and in this case, we confirmed it against cytogenetics. So traditional methods, in this case, the child sample was sent to India, cytogenetics were performed. In Accra, we did long-read sequencing, and we confirmed the structural variant that had been identified. So obviously, ONT is currently RUO, but it's showing the validity, and I think we saw a great talk today in the main session about the power of ONT in sort of leapfrogging across conventional. And the argument we make in Africa is, we're treating leukemia in Ghana without even doing flow cytometry. So I'm an immunologist. It sounds crazy to treat a disease without knowing if it's a B cell or a T cell. With ONT, we can go beyond just doing phenotyping, can identify mutations that are relevant, and we can identify structural variants. So we're looking at how we can now take this and adapt it to an accessible clinical application in a low-resource setting. And really, the theme of Yamachi is low resource does not have to be low-tech. Technologies like Oxford Nanopore allow us to be high-tech in a low-resource setting and effectively ensure that who we miss matters. Thank you.
Thank you so much. Fantastic. So before I open it up to the floor to ask questions, I'd like to ask a few questions of our panelists specifically. You and Prasad, you both showed really exciting work where we're leveraging ONT's ultra-long and T-to-T capabilities. And thanks for sharing that. Can you speculate why you think ultra-long ONT reads are particularly effective in fixing structural errors?
Okay, great questions. Okay. We'll know that the structural errors are critical for the improvement of the genome quality. And the longer reads can capture the long-range structural informations than the short ones. And when we use enough reads that longer than the structural errors, particularly large indels or some misassemblies like this, and the more if reads can span these errors, it is easier to detect and correct these structural errors, which can be maybe missed by the short reads. So particularly where there are many structural errors in the assembly, they may interfere with the short reads for their alignment from their original place, so that obstacle the identification and correction of the small errors. So I think that the capability of the ultra-long Oxford Nanopore reads is very critical for improving the quality of human genome, so that they can correct the structural errors at first. Okay.
That's great. Prasad, you have similar observations in
Yes.
the Singaporean samples?
What we have observed with the usefulness of the ultra-long data is mainly in the rDNA regions. Just to give one example, so what we found with the Indian genome, what we have constructed, so there was one SV in a repetitive regions. So that is present, or it's a fixed variant, you would say, a fixed variant within a South Asian population, but not present in the African or European genomes. So, but what makes it interesting is it's repetitive, it's hard to reconstruct. So with the ultra-long, we were able to reconstruct properly. And when checked, this SV falls within the ACMG gene list. So ACMG gene list is something which is clinically relevant.
Yeah.
So what you can see is with proper or accurate reconstruction of this region would enable us to see the SV that is present in existing genome that are European-based one, but it's a fixed allele within South Asian genomes. So yeah, this is one example where we could see how the longer read would help you reconstruct the regions very well.
Yeah. And that's a beautiful segue into the filagrant story, Francisco. In the asthma, atopic dermatitis genetics world for almost 2 decades, very early on, 2 loss-of-function variants were identified in exome 3 in the filagrant gene that affects European ancestry populations. We knew that African Americans suffered more from both atopic dermatitis as well as asthma. The one loss-of-function mutation is completely absent in African ancestry populations, and now this discovery in a filagrant gene. So my question actually to all 4 of you, but Francisco to start with, how do we move this information into clinical reference databases such as ClinVar and others to really begin acting clinically on these mutations that we're identifying?
Yeah, first of all, I think that particular locus needs to be much better resolved in not only in diverse genomes, but even in the reference. We have done local assembly that didn't show here, and we are comparing this to the T2T genomes, both the CHM13 or the AG002, and there's a lot of small misalignments happening, and what could be order structural variants versus those genomes, which tells me that we really don't understand the genetic variation in that gene because of the nature of the gene. So I think as we see more T2T assemblies, including looking at that region, we are going to start to make heads and tails of what's really going on there, not only in the structural variation, but also in the small variant. And the next challenge is how do we translate into disease databases, because obviously all of those are indexed to the build 38, so we have to do something creative there to be able to start using this in routine care. But definitely, I think this is just one locus. Imagine what else is out there.
Yep, great. I'll end with you, and then I'll open it. A beautiful summary of advancements that are being made in diverse populations in the clinical application of genomics, multiomics. So what are your thoughts about, you've been in the trenches here, how do we accelerate not just drug discovery, but the uptake of other clinical applications and beyond oncology in diverse populations? Are there lessons learned, challenges you've identified that would be applicable to other populations in parts of the world that haven't been as successful in building what you've built, for example?
I think we're still in the trenches. We're not out of them yet. I think from my perspective, a lot of the work on underrepresented populations has treated them as if they're only found in the countries in which they're underrepresented. Black people are only an underrepresented minority in Europe, parts of Europe, or in the US. And even there, the numbers are beginning to shift.
Yeah.
Black people are not underrepresented in Africa. So this idea that someone is underrepresented, it's just relative to where you are. An Indian person could be underrepresented in a Scandinavian country, but I guarantee you in India, they're not underrepresented. So the idea is we need to think differently about where we do this work. Historically, the technology has emerged from high-income settings, but as we're seeing in the emerged markets in India and Asia, you can see what that transformation can happen. Singapore is a giant now in biotech. What I think we need to do is understand that there is value in contextualizing our work. It's not just about having representation for the sake of it and trying to recruit minority populations because it's a good to do. It's about recognizing that when you study them in their environment, it makes sense. Because beyond the genome, if you think of the exposome, they are exposed to different things. The African genome is shaped by exposure to pathogens that are unique to the continent. So if you want to understand a person of African ancestry in the US, it helps you to link that data back to African individuals in Africa, so you can begin to distinguish what is admixture, what is exposure, ancient exposure versus current. So I think the real solution is to have a much more holistic understanding that genomics and precision medicine, you cannot just dissociate the individual from their context. And when we begin to do that, and we begin to recognize that great work can happen in places that have historically been ignored, that we can generate 30x genomes in Accra, and it will be comparable to something done in Oxford. When you begin to change your mindset, then we begin to unlock the real potential of these technologies. And I think that's the power of democratizing sequencing, but it's not about democratizing it in London. You know, it would be great if one day, instead of London calling, we do Accra calling, and we're all in Accra talking about this stuff, or in Lagos, or in New Delhi, or wherever. So I think that is how we need to think about it.
Here, here. Fabulous way to end that discussion. Beautiful. Thank you. Why don't we open it up to the floor? I think there are roving microphones. If somebody has, there's someone right here. Yeah, please wait till the mic comes to you.
Thank you very much, and to our last speaker, just following up on your patient recruitment, for all participants or patients, are you also getting baseline epidemiological information at recruitment, so that, to pick up on that last theme that you presented about pathogens potentially in Africa compared to America, I don't know, number of pregnancies, breastfeeding, things that are very important lifestyle factors in our current analysis. Is that also going to be part of your program?
Yes, short answer is yes. So we've actually built a cloud-based platform that we call Uvosio that allows us to collect standardized clinical data, so both for children and adults. The challenge we have, as you know, in many places in Africa is we don't have very good digital health records. So if you deploy some of these typical technologies here, they won't work. So we built this as a way to begin to digitize by asking targeted questions. So we design it for a particular cancer type, and we do it with the input from the local clinicians. And then we design these, and now, so for these 350 children, we have detailed longitudinal clinical data, including a family history, but then also what treatments they've received, how they've responded. So, because the metadata is really where the magic happens. The genome is nice, but you need the metadata to interpret it. So we are tackling that as well.
Fantastic. Any other questions? Yeah, right here.
So I think you mentioned in one of your talks that it's very difficult to estimate the base accuracy for the assembly, and you recommended looking at like the read alignments and all of that, but like it's very tempting to have like a single QV value representing the assembly quality. So do you have any recommendations how to do that? Like, do we use like orthogonal sequencing data? Like, what do we do if we want to estimate this difficult parameter?
For me?
You go ahead.
Yes, okay. You mean that use a different reference for the already whole genome sequencing data? Is that? Sorry.
Sorry, how do we estimate like base accuracy when we're getting to these like very highly accurate genomes?
The high dips in the coverage. The high dips coverage, how to handle these regions, you mean?
Just the base accuracy, like the Fred score, yeah.
Maybe I can take this one. So you are asking what is the best way to assess the genome quality, right? So as Professor Shi mentioned, and even we checked, we are taking more than one approaches to assess the quality, because your K-mer based, the difficulty is if a assembly K-mer is seen in the read, we just say it's good. But when we look at the longer structure, for example, beyond the K-mer size, if there is a misassembly, then it won't be picked up. because you still see the same K-mer signature, what you see in the assembly are seen in the reads. But the moment you look at the broader picture beyond that K-mer, then you see the assembly errors. So that's why what we do is we just don't rely on the K-mer-based approaches. We also do at the read-based mapping one. So that's where we find lot of structural variations. Even though you find your assembly to be 70 plus, right? But you still find structural variations using the same data set from which you have generated the assembly. So yeah, you can only use the K-mer approaches to call it as have a very high quality. But yeah, you would still have a lot of misassemblies, especially in the repeat regions like centromeres or RDNA clusters, right? So there, using multiple approaches would enable you to call your assembly as a high quality reference genome. Yeah. Yes.
So unfortunately, we're out of time. I want to thank all of you, the audience, for your participation, and a very special thanks to Prasad, Francisco, you, and Ya. I'm sure they'll be around to take additional questions. So thank the four of you for a fabulous conversation. Thank you.